So we did it, this one, meiosis and mitosis. Differentiation, redifferentiation, we have already understood. We have already discussed in plant growth chapter. So first come to the mitosis part. So mitosis is normal division, right? Suppose that we get wounded or any plant get plant part get damaged. So what will happen? The cells which are surrounding, they will start division. Suppose that I got a scar over here. For example, suppose that I got an injury or a scar over here. What will happen? There will be gap. The cells will get damaged and there will be a gap. But you know this gap will not remain as it is. So what will happen? This wound will heal and the gap will get filled. And after 1 or 2 months, you won't be able to know until unless you focus over here that there was a wound, right? So what will happen? The cells which are surrounding to this one, they will start dividing and they will fill the gap. When the gap will get filled, then the cell will stop, cell division will stop. The same process takes place by the same process is done by the mitosis. So everywhere where the cell get damaged or older cell die and the formation of new cell take place, in whole body part, the formation of new cell take place by the process of mitosis. Meiosis takes place only at the point where the gamete formation takes place. For example, sperm formation where the sperm formation takes place, where the egg formation takes place. So wherever the pollen grain formation takes place in plant, ovule formation takes place in plant, right? So in those places, meiosis takes place. So meiosis takes place only where the gamete formation is taking. The rest of the body is controlled by, or the rest of the body processes are accomplished by the process of mitosis. Try to understand what is the mitosis. This is simple cell division. When we say that the cell get divided into 2 and to 4 further, so it means we are talking about the mitosis. Right? Normal cell division means mitosis. So centrosome we have just discussed few hours back only. So centrosome is still in our mind, so you will be able to understand it more easily. So this is an animal cell. Why I'm saying animal cell? Because here the cell wall is not there. So the example which is PPT, which is visible to you, this is an animal cell. An animal cell, this is a nucleus, right? Nuclear envelope, outside a structured nuclear envelope. This is plasma membrane, this is nucleolus, and this is the chromosome. And this part in protoplasm, there is centrosome. Centrosome is there are made up of centrosome surrounded by a membrane, and there are 2 centrioles. They are at 90 degrees. We have already discussed that 9 plus 0 arrangement, 9 triplet of microtubule, they make a centrosome. Right? That we have discussed in detail, and I have told you what is the relevance of centrosome. So this is G2 phase. Now after the G2 phase, so interphase is start from G1, then S, then G2. After the G2, there will be G2M checkpoint. If the cell passes through the G2M checkpoint, then what will happen? Then the cell division will start. Look, what happened in prophase? Condensation will take place. Both of the centrosome, both of the centriole, they will start moving towards the opposite side. When they are moving toward the opposite side, formation of spindle fiber takes place. They are the fiber-like structure. Try to understand and ask me. Raise your hand if you are if you have any query, even in between. Right in the mid, if you get any query, then you have to raise your hand. Look, try to understand. Let me tell you. So these are spindle fiber. So formation of yellow color structure. You can see the spindle fiber takes place. And these both centriole, so centrosome, this outer membrane disappeared and they start moving to the opposite pole. In the same time, so this stage is called prophase. This is the initiation of the cell division process. Now, both of the centriole are moving toward the opposite pole. In the same time, you can see the nuclear envelope start disappearing, right? It will disintegrate. start disintegrating or degrading. Now what is happening? Now what is happening inside the chromosome? The unconsolidated means that this chromatin fiber will start shrinking. They will start shrinking. They will shrink down, right? And formation of sister chromatids take place, right? Again, what is happening? Formation of sister chromatid taking place. This is the pro-metaphase, right? Pro-metaphase. In pro-metaphase, the both of the centriole move reaches at the opposite pole. When both of the centriole reaches at the opposite pole, formation of spindle fiber takes place, and these spindle fiber attaches with the centromere. Look, this is the centromere, and I have discussed few minutes back that at both end of the centromere there is a plate, and this plate is formed of what thing? This plate is formed of a substance, and we know this substance is what? Protein. So this plate, this yellow color plate, right? This yellow color plate is called kinetochore. It is made up of protein, right? So this is protein structure and this kinetochore. So this kinetochore will make bond with the or spindle fiber will attach with the kinetochore. And what will happen uh, in this kinetochore uh, from here onward? So this is the centrosome. Now it's visible clearly. Now what is happening? These spindle fiber are attached with the kinetochore, and they will arrange these all chromosomes in a mid, right? So this was the pro metaphase. Then come the metaphase. The metaphase can be we can know that the metaphase, we can get to know that the metaphase, this is the metaphase, because all the chromosome arrange themselves at the equator, right? And when they arrange themselves at the mid or at the equator, so this is called equatorial plate or metaphasic plate. So by the help of metaphasic plate or equatorial plate, or we can say when all the chromosomes get arranged at the equator, this is called equatorial plate. Now we can know that this is the metaphase, right? Metaphase is the stage at which we can see the chromosome easily. Normally, the chromosomes are studied at metaphase. They are the most clearly visible at the metaphase. What happened? Now both the spindle fiber will start pulling these kinetochore in opposite end. What will happen? The chromosome will break from the mid. When the chromosome will break from the centromere will break from the mid, half of the chromosome means one part will go upside, one part will go back, downward. That stage is called anaphase. At the anaphase, the chromosomes are pulled apart. When they are pulling apart, try to understand what I have told you, that these metacentric will show V-shaped structure, submetacentric will show L-shaped structure, right? Remember, this acrocentric will show J-shaped structure, and telocentric will show I-shaped structure. So V shape, J shape, L shape, and I shape chromosome can be seen only at the telophase. Note it down, please. I'm sorry, I'm at anaphase. At anaphase, we can see V shape, L shape, J shape, and I shape chromosome. So they are the anaphasic chromosome. Clear? Because if they will be pulled by, right, then arms will make this shape. So after anaphase, both of the chromosome, group of the chromosome, they will reach at the opposite. Now, after reaching the opposite pole, these chromosome will again disintegrate. Means they will uncondense, right? So their visibility will reduce. Again, in the telophase, formation of the this nuclear envelope will take place. Nucleolus will reappear, right? Now, this was the karyokinesis. Nucleus have divided into two equal parts. Now, this is the time of division of the protoplasm of cytoplasm. I have told you that first karyokinesis will take place, then cytokinesis will take place. Karyokinesis means division of the nucleus, and cytokinesis means division of the protoplasm. Now, till this point, there is no difference in the division of the plant or animal cell. But in the cytokinesis means karyokinesis are almost same in both plant and animal. But cytokinesis is different. What is the difference between that? Try to understand. This was the animal cell. So what will happen? formation of a furrow will take place in the animal cell. So like this way, I have made the arrow. The formation of a furrow will take place. Furrow formation will take place, and then what will happen? This furrow will try to approach toward the center. It will become centripetal, right? And the cleavage will take place, and protoplasm will divide into two parts. So centripetal division will take place, and due to furrow, the cell will divide. Protoplasm will divide into two parts. Now again, what will happen in the plant cell? In the plant cell, deposition of the substance takes place at the center, right? The plant cell deposition of substance takes place at the center. From center, it will start expanding outside, right? It will start. Whatever the material deposit at the center, this is called cell plate, right? This structure is called cell plate. So what will happen here? The furrow formation will not take place. Plant cell, when the protoplasm will divide, the deposition of substance take place at the center, right? And the formation of cell plate will take place. The component which make the cell plate are called phragmoplast, right? Remember the phragmoplast. What is the phragmoplast? So phragmoplast is made up of microtubule, calcium, pectate, and all that, right? And they form the cell plast. Now, what will happen? This cell will from center to outside, means centrifugal fashion. This cell plate will spread, and this cell, when the cell plate will reach at the margins or um, at the cell wall, it will divide protoplasm into two parts. So in animal cell, the cleavage of protoplasm is centripetal, but in plant cell, the cleavage of protoplasm is centrifugal fashion, right? That's how the division of cell takes place into two parts, right? So this division was given by Sutton and Boveri, right? And Starsberger. Spindle fiber are made up of 4 to 20 microtubules, right? Tell me, guys, is it clear or not? Go through with it and tell me if you have any doubt in this. It is important. And this is conceptual. You have not to cram it. You have to learn it. Farhad, clear?
Yes, sir.
Great. So mitosis have been divided into 4 phases: prophase, metaphase, anaphase, telophase. That is called karyokinesis. Then protoplasm, that is called cytokinesis. So look, what is the phragmoplast? I have told over here, right? When the protoplasm divide in the cell, I have tried to show more clearly because sometimes it is not that much like students are unable to visualize that. So look here, the Golgi derive the vesicles, so vesicle get arranged, right? And the formation of cell plate takes place. Right? Then what happened? The cell plate formation is taking place, right? And all this material is called phragmoplast, right? Phragmoplast are made up of microtubules. Now the phragmoplast formation takes place. Now microfilaments connecting the phragmoplast. So these are the microfilament that is connecting the phragmoplast. This is phragmoplast. This is cell plate formation takes place. Now, after some time, the complete cell plate will be formed and cell will divide into. So this is called the cell plate formation or centrifugal division. Now come to the meiosis. Meiosis is a longer process. So meiosis have been divided into 2 stages: meiosis 1, meiosis 2, right? As I have already told you, in mitosis, the number of chromosome remains constant in mother cell and daughter cell. So mitosis is called equational division because the number of the chromosome remains equal in both. Right? Meiosis is called what we call meiosis? Reductional division. Number of chromosome get half. That is called reductional division. Look, now this is the meiosis. In meiosis, this is again uh, mm, cell in the animal cell. The same thing. So meiosis first, meiosis second is equal to mitosis. Meiosis first is a bit different. This is meiosis first. There, this is 2n cell, diploid cell, means 46 chromosome are there. This is centrosome. Again, in interphase, prophase 1, centrosome will start moving to the opposite pole. Formation of spindle fiber will take place. These structure which are visible outside, they are called ester rays, right? Now, what will happen? The formation of bivalent will take place. Now, try to understand. What is the bivalent? Bivalent means the chromosome will be in the pair of two, right? But in mitosis, there was single chromosome. Both chromosome have two sister arms, but chromosome were single. But here the two different chromosome are in pair, right? This is called, so you can see there is a one blue color chromosome, another red color chromosome. So two chromosome will come into pair, and this structure is called bivalent. So formation of bivalent will take place in meiosis, but in mitosis, bivalent will not be formed. There will be monovalent, means single chromosome with 4 arms. Here, there are two chromosome with 8 arms, right? You can see. Now, the same time in prophase, what is happening? You can see exchange of part of chromatid is taking place. This blue color chromosome is taking part of red color chromosome is taking part of the blue, right? Means they are exchanging their genetic material, right? This process is called crossing over process. So in mitosis, crossing over process do not take place, means exchange of part of chromosome do not take place in mitosis, but in meiosis, exchange of part of chromosome is taking place. Here crossing over is taking place. Why a parent have 4 child, all the 4 child have different faces. They do not until they are unless they are twin. Right? If they are not twin, their faces will not match. Why? Because at the time of gamete formation, crossing over takes place at the time of gamete formation in parent. That's why all child do not resemble dissimilar. This is the answer of this question. Right? We all are different because of this crossing over process, which takes place in human. Right? Look. Is that clear? Crossing over clear and bivalent clear? Guys, keep on telling me. Alan, clear? Crossing over and this one? Bivalent?
Yes, sir.
Okay. Hanifa, are you getting? Safullah? Getting it? Hanifa?
Yes, sir.
Yes, sir.
Very good. Very good. It is important. So crossing over is important. You have to remember. Bivalent is important. You have to remember. I'm stressing more on those points which are important for your medical examination. Then, what will happen? Again, the metaphasic plate, the chromosome, all bivalent will get arranged at the equator. We call it metaphasic plate. Now, this is the metaphase. Now, what will happen? Both of the chromosome will be pulled apart, right? Both of the chromosome will be pulled apart. So this diad will get, this bivalent will get separate, right? Now what will happen from one formation of, from one cell, formation of two cell. Again, cytokinesis will take place. And what will happen? Formation of two cell. Both of the cell is haploid. So meiosis 1 is reductional division. Why? Because in meiosis 1 itself, the number of chromosome become half. So that's why meiosis 1 is reductional division. Now every cell will have n chromosome, 23 chromosome. Now these 23 chromosomes are not getting reduced, right? Their number will not decrease, right? So here from 23, next the 4 daughter will have 23. So meiosis second is equational division, where mother cell, and now they are the mother cell. These haploid cells are the mother cell. Now the same thing will happen in prophase. Again, this microtubule formation will take place. Centriole will move to the opposite direction. In metaphase second, both will arrange in the metaphasic plate. And anaphase, both will be pulled apart. In telophase, both will reach at the opposite cells, and formation of 4 cell take place. So here in meiosis second, the formation of the number of chromosome is 23. After the meiosis second, number of chromosome is 23. So here the number of chromosome remains same. So what you have to do is you have to understand that you can get this question. Meiosis first is reductional division, and meiosis second is equational division. If you want to note it down, please note. Right. Is that clear? Go through with it and tell me, is it clear or not? If not clear, then ask me. Is that clear? Meiosis clear? If you have any doubt, please ask me. Feel free to ask me if you have any doubt. No doubt?
No doubt, sir.
Very good. Now I have one more PPT for meiosis prophase 1. So I want to tell you the so we have just studied meiosis 1, meiosis 2. In meiosis 1, prophase 1, beta phase 1, anaphase 1, telophase 1. Again, in the same meiosis 2, there is a prophase 1, prophase 2, metaphase 2, and all that. So I have told you that the crossing over takes place. So further, meiosis prophase is a very long phase. We have divided meiosis prophase into further 5 stages. First stage is leptotene. In leptotene, nuclear membrane, this is nuclear membrane, formation of this replicate into chromosome condense. Chromosome condense. In zygotene, formation of bivalent takes place. These two chromosome will come in pair, right? When they will come in pair, some of their part will adhere with the another chromosome, right? This structure is called synaptonymal complex, or the process is called synapses. It is written over there. This process is called synapses, and this structure is called synaptonymal complex. So, what is the question? When the bivalent formation will take place at zygotene, synapses or synaptonymal complex can be seen in zygotene. And next stage is packetine. In packetine, crossing over takes place, means exchange of chromatin arms will take place in packetine, right? The next stage is diplotene. In diplotene, you can see this cross structure because they are after the exchanging of the part, they are getting separate. So this Caesar-like structure will be formed. This Caesar-like structure is called chiasmata or chiasma, right? So chiasma or synaptonymal complex you can see over here in diplotene. So when the chiasma formation will take place in diplotene, synaptonymal complex, zygotene, crossing over in packetine, and in dikinesis, both nuclear membrane will break down and end of the prophase. So what happened in the crossing over? Look, these are the sister chromatid, these are the green color, these are sister chromatid. But this green and black are non-sister chromatid, right? So exchange of part takes place between non-sister chromatid. So suppose that, and here this is male, this is the chromosome of male, this is the chromosome of the female, X and Y chromosome. What will happen? Both will exchange their part, and due to exchange of their part, a new type of the chromosome will be formed, right? This is called crossing over process. And due to crossing over process, variation comes in the human, means the human faces become different, the body become different, the structure become different. So this was the prophase of the meiosis. Tell me. And what you have to remember, I have already told you. Whatever underlined, that is only important for you. You need not to learn anything else. If you want to write it down somewhere, you can. If you have any query, please ask. But chiasma, bivalent, crossing over, synaptonymal complex, these questions you will get. That's why I'm explaining everything with the help of diagram. Is that clear to all of you?
Yes, sir.
Now, what is the significance of meiosis? Formation of gamete takes place through meiosis. They maintain the number of chromosomes. That is very important. Look, in human species, parents have 23 pair, means 46 chromosomes. If meiosis will not take place, this 46 now they will become the pair of 4 pair of 23 in next generation. In further next generation, 8 pair of 46. In next generation, 16 pair of 46. By this way, number of chromosomes will keep on increasing, and the human might have become giant, like gigantic shape. But before the chromosomes are passed to the next generation, meiosis takes place. So generation after generation, the chromosome number remains only 2n or 46, right? So meiosis maintains the number of chromosomes in any species. Crossing over produces new type of the combination and variation, and that is help in the evolution as well, right? So this was all about this chapter. You have next 3 minutes. Go through with your NCRT and ask me if you have any doubt. Then we will move to the next part. Go through with your NCRT and ask me if you have any doubt. So I think clear to all of you. Must have gone through. Confirm me, then I'll move to next chapter. Hanifa, clear? Maimuna? I don't.
Yes.
Very good. Okay. It's clear to all of you. Fine. So before going ahead, let me open with that PDF where your syllabus, which I have committed to you. Look here, this is the syllabus, and what we did. Yes. So cell biology and cell cycle. We did cell biology and cell cycle. Next is animal and the plant classification, and then we'll after the animal and the plant classification, we'll finish our biology syllabus, right? So, but both are the bigger one. It will take some time, right? So let's start with the plant care classification type. Then we will discuss the animal classification part, right? So let's start with the plant classification part. By the way, you won't get that much question from the we have designed this syllabus, like I have designed this syllabus of biology like this way from the weightage itself, right? These are the chapter plant and animal classification. Do not read much more. Whatever I'll share with you, you have to read only that, because wasting time, that will be the wastage of the time, because you won't get like much questions, either one or two questions you may get from the plant and one or two questions from the animal kingdom. You won't get that. The previous 10 year back, these were very relevant. But still, these chapters are taught in every institute. These chapters, like NEET batches, start from the here, and these are chapters. They goes for the 1 month, right? Animal kingdom is taught for the 1 month, the regular batches. but hardly for the one question. They are just consuming the time. So don't waste your time. That is a information for you. So let's start with the plant classification part, right? So further, plant is classified. Plants are classified into two subkingdoms. So plant is a kingdom. So plantae kingdom had been classified in two parts: cryptogams and phanerogams. So what are the cryptogams? Actually, can you tell me what is the cryptocurrency? You must have listened this term, cryptocurrency.
Yes.
Yes. So what is the crypto term, Safullah?
I guess digital.
Digital. Very good. So Ellen will tell me. Ellen, are you there? Ellen is not there. Oh, Ellen, unable to. Who is tell me what is the crypto? Look, let me tell you in biology. Uh, you have told correctly, that's digital currency. So crypto is a Latin term. Crypto means hidden, right? And gami means marriage. Marriage, right? Hidden marriage. Actually, their sex organ cannot be seen. That's why their sex organ are not visible, clearly visible. That's why they are called cryptogams. You cannot see their sex organ and their. You can see the pollen grain, pistil, anther, and all that. And what are the phanerogams? Phanerogams are just flowering, right? Phanero is the visible marriage, right? So they are just flowering. So they are flowering plant, and they are visible, right? They are visible marriage, means visible sex organ, right? So that is the phanerogram, and that is the crypto. Okay, so now you will remember why I am breaking the term. I always break the term because it give a good imprint on your brain, and you will remember that what is the cryptograms. You won't get confused. So always break the term while reading. Cryptogam, so subkingdom, cryptogam, and plerogam. Further, cryptogam divided into three parts: halophyta, bryophyta, and pteridophyta. Can anybody tell me what is the thallophyta? The definition is there. There is a thallophyta. Thallophyta. Let me tell you. Thallophyta means thallus-like body. Thallus-like body. What is the thallus? Thallus-like body. What does it mean? It means, oh, where we will use the term where. So thallus will be called those plant body, the plant body, plant body where root, stem, and leaves cannot be they are present, cannot be differentiated easily. Okay. There are not root, there are root like structure, there is not stem like structure, there is not leaf like structure. So those kind of the plant are called thallophyta, right? Bryophyta, I will tell you, bryophyta have some differentiation of root and leaf like structure. Pteridophyta, there are proper leaves, root, and the stem are there, right? So further, thallophyta are algae and fungi. Algae and fungi, there is a no root, no stem, no leaves. You cannot use this term, right? Then bryophyta, there are leaf-like structure. There are root-like structure. There are stem-like structure. And pteridophyta, there are proper root, stem, leaves, but there is a no seed, right? In spermatophyta are those which bear seed, right? Bear seed. So seeds are present. But further, the spermatophyta are two types: gymnosperm and angiosperm. In gymnosperm, gymno means naked, and sperm is seed, right? So in gymnosperm, there are naked seed, naked, right? And angiosperm, there are covered seed, right? So angio means covered and sperm means seed, right? So that is a classic plant classification. Did you got that classification? Clear? Halophyta I have written over here. Halophyta, that is the lack of well differentiated body structure, and the plant body is thallus. Right? Thallus, I have told you what is the thallus. Halophyta includes the plant with primitive and simple body structure. Then the plant body is thallus. They may be filamentous. So what kind of plant body may be filamentous, like thread-like structure? Plant body may be colonial. It may be branch structure. It may be unbranched structure. Example include algae, red algae, and brown algae. We'll discuss algae in detail, right? Hope the plant classification might be clear to all of you, right? So first we will start from the Thallophyta. In Thallophyta, we will start from the algae. What we are going to start in the plant kingdom, fungi have not been included in the plant kingdom right now, right? We do not study fungi. Fungi is a separate kingdom, right? So first, sometime we include some classification, some classification do not include. So normally in NCRT, in our classification system, what we are going to study, we do not include fungi, right, in plant. So, but they are a thallus-like body. So first algae. Second, we will study the bryophyta. Third, we will study the pteridophyta. Fourth, we will study the gymnosperm. And fifth, we will study the angiosperm. So let's start one by one. First, we will start with the algae. So what are the algae? Is that clear to all of you? Prat, clear?
Yes, sir.
Very. So let's start with the algae. Wherever you guys feel that there is some ambiguity or you are not getting clearly, raise your hand. Feel free. So algae are like it's a thallus-like body. You cannot tell that it's a root is there, shoot is there, or let me show you any example of any algae. Then you will. By the way, you must have studied it, but still, let me show you some diagram of algae. Look, this is an algae, Spirogyra, right? In Spirogyra, you cannot tell where is the root, where is the leaf. Can you tell me? No, there is not. You won't be able to. This is thread-like structure. This is algae, right? So here, root, stem, and leaves cannot be defined, right? You cannot differentiate between the root, stem, and leaf, right? So this kind of these are just made up of the cell, and they are a thread of the cell, and this is a Spirogyra. So it will look. It will have all structure of the plant, like you can see. uh, Look, here you can see that all the structure of the plant are present, right? You can see this is cell wall is present. That is a characteristic of plant. Vacuole is present, plant cell, right? Cytoplasm is present, obviously. Then there is a chloroplast. So chloroplast is very peculiar in the algae. Look, this is not chloroplast like that chloroplast which we have studied in higher plant. That look like a ball of the rugby, right? Means that is a discoid shape. That kind of the chloroplast are not found over here. So what kind of the chloroplast? This is the ribbon shaped chloroplast. So it's a ribbon shape. And inside the chloroplast, you can see the pyronoids. I have already told you what are the pyronoids. Pyronoids are the characteristic of algae, basically, right? And pyronoids are the food storing body. Food storing body. They are found in algae. What can be food? Food can be anything, right? Food can be, what can be food? That can be carbohydrate. That can be carbohydrate. Uh, that can be protein. That can be fat, right? So that can be anything, can be the food storage, right? So food is stored in pyrenoids, and pyrenoids are remains stuck or remains sunken within the chloroplast. This is peculiarity of the algae that you have to remember, right? Chloroplasts have different shape, and outside of the algae they have mucilaginous sheath. Mucilaginous sheath, it protect them. It is a slimy layer, right? And it protect them from dehydration. Protect from dehydration. They dehydration. They keep them hydrated, right? And this is cytoplasmic strands are found inside and nucleus is present inside, right? So this is the structure of algae. They do not have root, leaf, or stem-like structure. Further, algae is divided into three parts, right? Chlorophyce, Pheophyce, and Rhodophyce. Chlorophyce, this is the class of algae, right? So there are three classes of algae. There are three classes of algae. These three classes are chlorophyce. In chlorophyce, we study green algae. They are green in color. Green algae. This is the most advanced algae, right? Then pheophyce is called brown algae. This is called brown. Brown algae, they are brown in color, basically marine. And rhodophyce is known as the red algae, right? Red algae. This is red color algae. So there are three kind of algae. Look. This is already, this must be given in your NCRT. I think it's there in your NCRT. So this is very, right? Look. Chlorophyce, when we talk about the classes of algae, so there are three classes of algae. So first class of algae is chlorophyce. Chlorophyce, I have already told you, this is the green algae. In green algae, this is green algae. It will have chlorophyll A and B. So chlorophyll A and B pigment will be found. The food is stored in the form of pyrenoid, right? So I have already told you, food will be stored in pyrenoid. In pyrenoid, food is stored, food is starch. This question is normally asked. And their cell wall is made up of cellulose, right? And they may have flagella, 2 to 8 flagella, and they are found in the fresh water, right? They are found in the fresh water. They are also found in the salt water, right? So fresh water and salt water both they are found, right? They are marine. That means they will be found in the rivers, lake, and as well as in sea, oceans. Pheophyce is called brown algae, right? It will have chlorophyll A and C. Look, chlorophyll B is the chlorophyll which give green color, but here chlorophyll A and C is found. You have to remember fucoxanthin in found. Fucoxanthin give brown color to these algae. Fucoxanthin give brown color to, it is a brown color pigment. It gives peculiar brown color, impart brown color to these algae. That's why they are called brown algae. Second thing, what you have to remember, guys, whatever I am saying, that is very important. These are the question only. You will get question from here only, and you can see that I'll give you the in practice set as well. I'll give you these kind of the question. Look, the stored food in the parentoid. What is mannitol? This is carbohydrate and laminarin, right? So mannitol and laminarin is the compound which in which form the food is stored. Starch is carbohydrate, mannitol and laminarin is also this one, right? Starch, carbohydrate. So the cell wall is made up of cellulose but and algin. Remember algin. So algin is a substance. Can anybody tell me what is the use of algin? Any one of you? Is there any smart?
It is used in jellies. Okay.
Very good. And any more practical thing? Algin is used in killing. Look, very important thing. When you take the medication, you take the capsule. When you, as you put the capsule in your tongue, it get moist and it burst. Right. Put a capsule in a water, in glass of water, it dissolve immediately. That capsule, the wall of the capsule are basically made up of algin. Right, that is algin. and obviously jelly, and those artificial fruits which you put in the ice cream, they take the taste of ice cream. They all are made up of algae, right? So that is the use of algae. And look, here the flagella are equal. 2, 4, 8, flagella will be here, flagella. Here the flagella will be unequal, right? So what does it mean by equal flagella? Equal flagella means suppose that algae, this is algae, this is a Clibodomonas actually. This is a green algae. They will have two flagella and both flagella will be equal in the length. Now here it is given that there will be two unequal flagella and they will be lateral. What does it mean? It means suppose that this is the gamete of algae. It will have two flagella. One is smaller, another is a larger one. Right. The larger one have these hair-like structure. Right. So there are two flagella and both are unequal and lateral flagella. This is the front flagella, right? Means vertical flagella or apical flagella. This is lateral flagella, right? And the third thing, flagella is absent in red algae. That is also question. I have seen that question number of times, right? So that is flagella are not found in the brown algae, and they are stored food, low radiant starch. That is the stored food, low radiant starch. This is also starch, right? So you have to remember the food, and then the cell wall is made up of pectin, pectin, right? And polysulfate esters, right? They are also fresh water and also found in the salt water. Look, here you will see that this is also found in the fresh water, but most of them are marine, right? So rhodophyci, they are mostly marine, mostly marine. They are rarely, some exceptions are there, mostly marine. Means they are mostly will be found in the oceans. Tell me the common characteristic of algae is clear or not? Then I will start one by one. Chlorophyll. Hanifa, clear? Maimuna? Hiba?
Yes, sir.
Hanifa and Hiba? Hiba, I don't listen you.
Yes, sir.
Very good. Hiba, what's about you? Please tell me.
Yes, sir.
Okay, very good. Aksa, clear? Getting it? It's easy, by the way, but the thing where I have made the star, you are getting the link of PPT? Getting the link of PPT or not? Tell me.
Yes, sir.
Okay. Whenever you do not get the link of PPT, let me know. And whatever I underline and make the star, it means this is, you have to cram it at any cost. This is the question. You know, every day, at least I make 200, 400, 500 question every day for the test. You know, and we have gone through with the lakhs of the questions, lakhs of the question. You know, so whenever I teach you, whenever I encounter anything, it recall me that how many time I have seen this question. We always go through the previous year question paper, and we do not give this task to our team to that, so that they select the question and they make the question. They can do, they all are the masters. All those who are typing, making the question paper, making the test, evaluating, they all are masters, at least masters from the same subject. But we do not give this task we always give to the senior teacher, so that we make the test which is helpful for you in examination. And we always after getting print and all that, we always review that, right? So that's why I am telling you, when I make the star, when I make the underline, it means I have seen that multiple time, or this is very important, or this kind of the question have been asked, this can be asked, right? So at least cram all those things. Do not leave anything which I have underlined or make the star. So we'll tackle one by one algae. First, we'll study the class chlorophytes. Green algae, right? The class chlorophytes. So first, I'll show you this algae. Actually, the chlorophytes is represented by this algae. The name of this structure, this algae is, it look like insect, but it is not insect. Can you tell me which algae is this one? No one? This structure belongs to the name of this green algae. It is a green algae. And its name is Chlamydomonas. Chlamydomonas. Chlamydomonas is a green algae. It's a green color algae. And this is representative of chlorophyll, right? And the Spirogyra, which I explained you commonly, that was also a member of green algae. Green algae are the most advanced algae, basically, out of all these algae. The oldest one is the red algae, then evolution taken place, brown algae came, and after the brown algae, green algae came. So green algae are the most advanced. Look, they have flagella. Rather than cilium, call it flagella. So this structure is called, rather than cilium, call it flagella. So this is flagella. Then flagella come out of a point and the root that is called basal body. So flagella arise from the basal body. Then there is a chloroplast. You can see this structure, this green color structure is chloroplast. Chloroplast is cup shape. So in cup shape, that you have to remember. Cup shape chloroplast is found in the Chlamydomonas. It's a green algae. So chloroplast is cup shape. There is eye spot. This eye spot work like basically eye. This is photosensitive. This is photosensitive sensitive. This is photosensitive structure. It is photosensitive structure, right? Here we can sense with the light, right? It's a photosensitive structure. Now, this is cell wall. Cell wall is made up of cellulose. This is nucleus. They have Golgi body, starch granule. You can see mitochondria, plasma membrane, and other important pyronoids. So these are the pyronoid. You can see this cup-shaped chloroplast, and it have the pyronoid. Pyronoid is the food storage body. So food is stored in the pyronoid structure. Food. What can be? This will be starch. We have studied, right? So pyronoid will be storing starch. Again, there are small volvox in this structure. This colony is found. So number of the volvox, they are the volvox, right? They look like the this one Chlamydomonas, but this is structure of volvox, right? Volvox. This is a colony of volvox, right? This is colony of volvox. A colony of volvox may be made up of, these cells are not interconnected with each other, right? They are just connected with each other, but they do not, they are individual cell, every single cell, right? And they have the two flagella. The colony of volvox may be made up of 500 to 60,000 volvox cells. 500 to colony of volvox might be made up of, it may be 60,000 cells of volvox, right? And these are the dot, and they have the flagella, and this is mucilage or gel-like structure. There will be 500 to 60,000 cells, right? And this colony is known as synovium. Synovium, synovium or coinovium, but normally we call it synovium. So this is representative of chlorophyll or green. Is that clear?
Yes.
So what are the characteristics of green algae? Let me write down some points for you, which are important, or the point which will be beneficial, or they can be asked in your. So let me write it down some important point. So these algae are mostly aquatic, means mostly they will be found in water. Mostly aquatic, right? And they may be both, right? Maybe marine or freshwater, right? Marine or freshwater. Means they will be found in oceans, they will be found in lake both. Fresh water. That was the first point. Second important point is that the plant body is thallus-like body. Thallus-like body. Thallus-like body. That I have already told you, but still let me write it down. That is no root, stem, or leaf. These structures do not exist. Right? Then the third point. The third point is the body is covered by mucilage, right? So it do not avoid, it protect them from desiccation, right? So body covered with mucilage. Then vascular bundles are not found in algae, any algae, right? So vascular tissue means xylem and phloem. Vascular tissues, let me write more clearly. Xylem and phloem. Xylem, phloem are absent. Are absent. So vascular tissues are not found, they are absent. Mechanical tissues means anything which gives them support, they are very soft kind. So sclerenchyma kind of the tissues, like woody structure, that is not found, right? So mechanical tissues are absent. Mechanical tissues are absent. Tissue are absent. Hence, plant body is flexible. Plant body is flexible, right? So they have flexible plant body, right? Next point. Grana are absent in chloroplast. They are photosynthetic. Photosynthetic. And grana are not found. Grana, very important. Grana are absent in chloroplast, right? So chloroplast will not have grana. Chlorophyll A and chlorophyll, that we have already understood, right? They will have chlorophyll A and carotenoid and xanthophyll. So that we have already discussed previously. Asexual and sexual, both kind of the reproduction is found. Asexual and sexual, both the reproduction is found. So both kind of the reproduction, asexual as well as sexual reproduction, found over here, right? Very, very important. Embryo is not found. Embryo stage is absent. That is a question. Embryo stage is absent. Embryo are not found. Clear? So one last important thing. What is their use? Is there any economic importance? So economic importance from, you will get the question from economic importance. What is there if it is there? Economic importance. Economic. So what is their contribution? So this is very, very important. You have to remember that chlorella. Have anyone listened this name? Chlorella?
Yes, sir.
What is their use? Chlorella and spirulina. And spirulina. First, I want to tell you spirulina. Spirulina. Right. They both are known as space algae. Space algae, because they are used in space. What is the function of spirulina? Spirulina is used as food supplement. Nowadays, people are taking spirulina capsule, food supplement. 1 teaspoon of spirulina is sufficient for the providing all nutritional value for 1 day to like for 12 hours to a human. Like it is that much. It is the superfood. Nothing is more nutritious in this earth than spirulina. Right. Then nowadays people are making a tablet of spirulina. Another is chlorella. Chlorella is used for purifying the gas in the space ship. Right. So it purifies gases. It absorb bad gases and provide good gases. Right? It is used as a superfood. Remember, so both are known as the space algae, right? And purify gases and decomposition of sewage treatment. Sewage treatment. Chlorella is also used as treatment. You can get question from here. It is very. Let me make the star. It is very important. Right? Is there any more? Yes. Algin is obtained from, there is a component called algin. We were discussing that. Algin is obtained from this. No. That is chlorella and spirulina are the green algae, rest are not algae. Algin is not extracted from the green algae. One very important question was there. Just a minute, give me 1 second. Red one. There is a disease, and that disease is yes, Cephallurus. That is very important algae. You can't forget that algae. Cephallurus got the red rot disease of the tea, red rust disease. So there is a parasitic algae, parasitic genus, Cephallurus. Cephallurus. Cephallurus is a parasitic algae. That question has been asked multiple times. Parasitic algae. By the way, I want to tell you, algae are rare in the rarest case parasitic. They are symbiotic. They are not parasitic. Very few algae are parasitic. So this is parasitic algae, and this causes uh, a red rust disease. It causes rust disease of tea and coffee. That you have to remember. Tea and coffee. It causes rust disease of tea and coffee and reduces the productivity of tea and coffee. Right. And what else? Food is a star that we know. Eye spot is present that we have discussed. Ball box, I have told you. Right. Spirogyra. Both asexual and sexual reproduction is found. Chlorella, then that we have discussed. Cephallurus, the red rust of the tea. That's it, right? So this was all about the green algae. And what you have to do, you have to remember all these points. Is that clear? If you have any doubt in any point, please ask me. By the way, this is Spirogyra, which we discussed. Spirogyra is known as water silk. That you also have to remember, because it causes it grow very fast in the lake and it pollute the lake, right? So Spirogyra. I have already told you Spirogyra. It causes the water bloom. Remember algal bloom, right? So Spirogyra is known as the water silk. We have already discussed in the ecology part. water silk. So, and it is responsible for algal bloom in lake, algal bloom in lake, right? That we have discussed. So tell me, is there any doubt in green algae? It's part of the green algae. Then we will go ahead with the brown algae. Tell me if you have any ambiguity or any confusion in what, ask me. Clear? All point clear?
Yes, sir.
Okay. So let's start with the next algae, and the next algae will be brown algae. Right? Brown algae. So brown algae is known as pheophyce. And first, let me show you some picture of the brown algae. Brown algae. This is one diagram. Let me insert some more diagram of brown algae. So these are the diagram of brown algae, right? And these are the largest algae. They are the giant algae. You can't even imagine how longer they can be. Brown algae can be up to uh, brown algae can be up to 100 meter, right? So this is the giant algae, and this is the diagram of laminaria. Laminaria is one of the most uh, one of the giant algae. It can be up to 100 meter in the water, right? Now you can imagine how giant it is. And all the iodine which you are iodine is not found in the here on the earth, right, as a mineral. Iodine is extracted from the laminaria only. So iodine which you are eating in the your this salt. So this iodine is coming from the laminaria only. One more important thing, they have a root-like structure. It is not a root. Again, thallus-like structure. So this is called hold fast. This is structure is not root. It's a hold fast, right? This is a type, and they have some air bubble-like structure. They help in the buoyancy. And these leaves are not called leaf. Basically, they are called blade. Right? And again, it is written. This is the thallus-like structure, and they just reproduce. They produce these spores, right? These spores are male and female gametophyte. They produce sperm and egg. They fertilize. After fertilization, again they make a new plant, right? So that's how they do. So this is the brown algae. Now, what are the characteristic of brown algae? Let me discuss with you one by one. So, common characteristic of brown algae. So they possess. So first and foremost feature is that they have their color is brown. So brown color is due to fucoxanthin, right? So they possess chlorophyll A and chlorophyll C, chlorophyll C. Then fucoxanthin, fucoxanthin. This fucoxanthin gives them brown color. We have already discussed that, right? And the reserve food is laminarin that we know we have already discussed in the starting of the class. And majority of them are marine. Majority of them are marine, right? So they are marine algae basically. They are found in salt water. Right? One more important thing, they are also called kelp. Brown algae are also called kelp. Kelp is a term. Kelp stand for kelp means CV. That is, they are called CV, right? CV. They are known as the CV. And next, they do not have unicellular form. Means unicellular brown algae is not found, right? So unicellular, that is a very important question. Let me make a star over here. Unicellular forms are absent. Unicellular forms are forms are absent. So unicellular forms are not found over there, right? Then they are also known as the kelp that I have told you. And brown algae maybe there are two brown algae. So largest brown algae is macrocystis. So macrocystis, that is the largest brown algae. Largest kelp may be up to 40 to 100 meters. Maybe. Can you understand? 40 to 100 meter means approximately 350 feet. Maybe up to 40 to 100 meter in length. Right? Nereocystis maybe 20 to 30 meters. Nereocystis maybe 20 to 30 meter in length. Right. So microcystic, nereocystic, you have to remember they are the largest brown algae or largest algae. Right. So body is divided into, so plant body is thallus-like body. So thallus is divided into three parts. We have understood. Thallus is divided into root-like structure that is called hold fast. Right. Hold fast. Then stipe. Stipe is a stem-like structure, and blade that you have already seen in the diagram. Right. Right. And they have the air vesicle, right, or air bladder that provide them buoyancy, right? That is the largest algae. How they stand like this way, right? So ribbon-like structure. So they have air bubbles, right? They they often possess air vesicle, air vesicle, right? These air vessels or bladder or bladder where the air remains filled. They provide buoyancy, provide buoyancy, right? That you have to remember. They have air bubbles. That question I have seen. And what else? What is economic importance? Chlorophyll, we have fucoxanthin give them a brown color that I am telling again and again. And so they have both sexual and asexual reproduction present. Asexual reproduction is found right? Let me see if there is any economic importance, then I will tell you what is the economic importance. There any economic importance? Yes, we extract bromine. Yes. From laminaria. From laminaria, we extract iodine. That is very important. Iodine and bromine is extracted from here. Bromine extracted. So we get bromine and this one, bromine and iodine from the laminaria itself. And is there any more economic importance? This was the economic importance, right? And we'll start the red algae in the next class onward, right? So next is the red algae, and you have to remember that we have to start from the red algae onward. Go through with it and let me know if there is nothing much more to understand, but you have to remember the what you have to remember. You have to remember the food stored. You have to remember the economic importance. You have to remember the that what kind of the pigments are found, chlorophyll A, B, C, fucoxanthin, right? That you have to remember. And if there is any peculiarity, then exception, like this is we extract. This is the largest kelp. It may be up to microcystic, maybe up to 100 meters. So that kind of the fact you have to remember. So you have to remember almost all of those things, whatever I have written over here, right? And ask me if you have any doubt. Tell me. In next class, most probably we'll finish this one, uh, plant classification, because plant and animal classification both are the very big chapter. So at least they will take one class minimum. Any doubt? Any question? Yes. Tell me if you have. No doubt. No question. Ask me if you have any doubt, any question.